Wet mount is a technique for one to employ to achieve the desired observation result given the specimen and the specific part of the specimen that are of interest to the observer. In a wet mount, the specimen is suspended in a drop of liquid, located between slide and cover glass. It is a common slide preparation technique used to observe many types of live microorganisms. Wet mounts cannot be stored over extended time periods as the water evaporates. For this reason, a wet mount is sometimes also referred to as a temporary mount. Wet mounts can be generated using a variety of liquids such as water, immersion oil, and glycerin or glycerol, but water is likely being the most popular.
Hey guys, I'm Gina from BYFT1 and we are here today in the laboratory with my groupmates to demonstrate the wet mount technique.
First and foremost, it's kind of important that you mix samples a little bit so that to make sure that all the algae are properly suspended in the water. Next, use the dropper to place a small droplet of pond water in the center of the slide. This should be a very small droplet. Then, using the tweezers, take the slide cover slip and place one side of the cover slip down on the slide with the other side still above the slide. If you have added too much water and the cover slip has floated, use the edge of the paper towel and place it next to the edge of the cover slip and just let the paper towel extract some of the excess water. Now you are ready to take your wet mount slide and place it on the microscope stage and start observing your specimen. You can see in this observation some microorganisms living in the pond water where the specimen is taken. The main aquatic groups include arthropods, bacteria, protozoa, hydras, fungi, phytoplankton, and algae. If the specimen is not in water just like this onion, first, place a small drop of water without specimen in the center of the microscope slide. Place the specimen into the water. And add some more water on top of the specimen and make sure that the specimen is completely submerged. Otherwise, there is this possibility for air bubbles forming between cover glass and specimen. Hold the cover glass on one side with the help of tweezers. Lower the cover glass onto the water drop at an angle. Then, slowly lower the cover glass into the liquid. This will minimize disturbing air bubbles. Lastly, remove excess water with filter paper or tissue paper. The cover glass should not float freely. The surface tension of the water should hold it in place. Alternatively, you can add more water using a pipette or tweezers. If the specimen is dry, specifically the onion, you can observe the thin membrane rectangular interlocking cells, visible distinct cell walls, nucleus, vacuoles, and small granules inside the cell's cytoplasm. Compared to permanently mounted slides, wet mounts do have certain advantages. Quick preparation. Specimen fixation, dehydration, and staining are not necessary, but possible if required. For this reason, wet mounts are the first kind of mounts that students learn to make. Few artifacts if there is no chemical and physical processing of the specimen before observation or no fixation, there are little artifacts and the specimens appear in their natural condition. Living and moving. It is possible to observe living and moving organisms. It is also possible to observe certain processes of life, such as feeding, cell division, etc., for water-based mounts. Natural colors. The colors are natural and not faded. The colors of permanently mounted specimens may fade over time. Disadvantages of wet mounts include movement. The advantage of observing movement can also be a disadvantage. Due to the movement of the organisms, it may be more difficult to take pictures or to make drawings. There is a solution to this problem. One can slow down ciliates and other protozoa by adding a solution such as ProtoSlow, which increases the viscosity of the water. Evaporation. The heat of the lamp causes the water to evaporate more quickly. More water must be added under the cover glass from time to time. Focus. Some organisms may swim vertically in the water and therefore move in and out of focus. Here it is important not to use too much or too little water. Too little water may squeeze the specimen between cover glass and slide. Storage. Wet mounts cannot be stored over a longer time.